aware of. I'd like to pivot now to the last part that I want to talk about, which is now the unconscious hippocampus. So we talked a little bit about the conscious hippocampus, and this was our prominent model. So we focused on this conscious aspect, right? And we just talked about this, and we're adding a new branch here. But now we're going to move beyond recollection familiarity and talk about this part, the implicit memory, right, of our canonical traditional model. And when Eric Kandel wrote his book, he tapped into the dogma that many others have introduced before he did. But the idea is that implicit memory does not depend on medial temporal lobe structures. Okay? Classic dogma. Absolute. Every textbook around. So, and this is sort of a representation of how people have thought implicit memory is served by different neural structures. So particularly the sensory and cortical areas. So the frontal cortex is supposed to be involved with conceptual priming, the repetition priming, this sort of superior temporal lobes, and the perceptual priming, and more the sensory cortices for visual information. But some alternative views have been developed, though, that have proposed that the hippocampus might play a role in some non-conscious processing, but more or less for the associative role of binding information together, and less so for the memory. So we know that there's this basic model, but there can be some exceptions. There's some preliminary fMRI evidence that came out of Roberto Cabeza's lab at Duke, as well as Larry Scar's lab in San Diego, that's found that the medial temporal lobe can be sensitive to the study status, basically whether a word was old or new. So it can show some memory-related activity, regardless of whether or not the subjects actually detected those differences. The hippocampus has been found to be related to implicit associations using some forms of fMRI, and notably eye tracking measures, which are a really powerful way to study a non-conscious memory because your eyes are incredibly sensitive and it has the temporal resolution that EEG has as well. So eye tracking has been very powerful, but what's lacked has been EEG studies of patients who have these specific deficits to show a necessary role. And it's possible that this wealth of data since 1957, even going back beyond that to Korsakoff's patients and beyond, they've only used really behavior and fMRI measures. And so it might be that two-dimensional approach for a 3D picture. And we might not be using the sensitive enough measures to detect differences that might truly be there. So we know behavior is very coarse of a measure. We certainly know fMRI is very coarse of a measure. So none of these studies have used physiology on hippocampal amnesia patients, and that's something that I wanted to approach, following from Eric Kandel and these other ideas that what if the hippocampus is playing a more fundamental role in implicit memory, perhaps even a necessary one. So how do we capture unconscious memories during a conscious memory test? It's kind of a problem, right? So it's the answer to that is similar to the answer to the question, how do porcupines mate?
Very carefully.
Yes, very carefully. All right, so the way we can capture non-conscious memories during a conscious memory task is very creatively and with EEG. You've heard that joke before. I told it in class. So the idea is physiology provides us a lot more leverage to gain insight here. So what you can do is you want to look at brain activity for the times in which people report that they have no conscious memory. So times when they forget. You show them an old piece of information. You say, hey, do you remember this? And they're like, nope. So we call that essentially a miss, right? So when you do that, that's this top line here. Previously seen but unrecognized test items. Now, what you do then is you compare that physiology to the times in which people report the same exact conscious experience to you. They say, nope, I don't remember it, but this time they're right. So this is on the lure items, the new piece of information, right? And that's going to be so this gives you your sort of your control, your baseline comparison condition, and the subjects have told you the same thing, identical, right? So the physiology should also be identical. If it's not, you're not able to attribute it to a conscious factor. There has to be something that the brain is remembering that the mind has forgotten. And that's if you see a difference, any difference between the physiological traces, which are statistically reliable, it shows you that something in the biology system is detecting and can remember something that is not accessible to conscious awareness. All right? Pretty cool. Okay. Can I get a question? Maybe? Okay. And so, again, a couple of points about this. Timing is really important. About 400 to 600 milliseconds. If we want to replicate this, if we want to use this as a tool, a dependent variable to look at in patients. And the obviously this is the physiology is important. So this is capturing something that behavior might be insensitive to. And so the idea is, can we adapt this approach for our confidence ratings? Because confidence ratings give us a whole lot more leverage on our conscious experience, right? Because we can basically quantify the different gradations of it. So how can we do that? All right? Well, again, sort of the way porcupines made, very carefully. So it turns out that there was another paper that had been published very recently, actually from a graduate student who had recently graduated here, Sarah Yu. I don't know if any of you guys know her. She's gone on to do wonderful things with Nielsen neuromarketing since. But she published a paper in 2010 where she also used an approach that did this in healthy, normal people. And it was this obscure analysis buried in some paper that I had read, and nobody could really even know that they did it. It was buried in there. But I came across it, and the way that we normally look at explicit memory, so we have our confidence ratings, 1, 2, 3, 4, 5, and our old items and our new items, and you can see the different distributions of how many times people say each response. And so we get our explicit memory contrast, right? Times when they got it correct versus wrong, and we can see that mid-frontal effect related to familiarity. We've talked about that today. So we want to recategorize this information to get that non-conscious comparison. The way that you do that is pretty interesting. You take you want to equate. You want this and this to be exactly equivalent for their conscious reports. And the way that you can do that is make sure that the confidence bins going into this are equivalent as well. So what you do is you identify the least amount of trials. So in this case, 7 is less than 19. 5 is less than 28. So you randomly select 5 of these 28 and randomly select 11 of these 54. You average across. So visually, the way that works is you end up with the same amount of trials in each consciously reported condition, and you average those across to create a condition of old information. You'll average these across to create new. Now what's critical about this is you've absolutely equated for their conscious experience of what they've told you they can declare. Right? And so I know it's a little bit more complicated, but if it's easier, rest on the slide beforehand where we compared the misses to correct rejections. And just know that conscious experience is held constant here between them. And anything that differs, because it should be the same, if it's different, then it means the brain is remembering something the mind has forgotten. And when she did that, she found posterior activity during the identical time period that there was frontal activity for conscious memory. So we know that they're dissociable during the same time. So if you're a detective and you're trying to find out who is the real perpetrator who robbed the bank, then turn that off. And you have two suspects. One of them has an airtight alibi. It means that he cannot have done it because he was in a different place at the same time. These patterns of activity are in different places at the same time. So they cannot be the same type of activity. Conscious, non-conscious, dissociated. Let's see if it changes based on having a hippocampus. And so what we did is I recategorized that data that I showed you earlier with the hippocampal patients. And just to remind you, this was their damage. And we want to know now about implicit memory as opposed to their explicit memory. And it turns out, this was the paper from Sarah Yu here, famous UTD alum. And first, I needed to know if in my paradigm, can I replicate what she found in a group of healthy 25 people? So that's what I did. I found my mid-frontal effect, completely replicated that these are the control subjects. I have 6 healthy age, sex, gender, education, IQ matched controls. And our hippocampal patients have the same familiarity-based effect. That's pretty cool replication. 4 studies, 2 laboratories. That's tight. Right? The action, though. Right? Here's the implicit memory. Can we replicate this? Will this be here? Will it be here? And it should be here, right? Because why wouldn't it be? Patient HM said they're fine. We've known this since 1957. It's also one of the reasons nobody's really looked for this stuff, because why would you? Right? How many of you guys think you could convince your PI to fund a project to show something that you guys already know? All right? It's kind of tough. Well, we replicated very nicely for our implicit memory effect in our healthy 25 subjects. Replicated again beautifully in our healthy age-matched controls. It's completely abolished in patients who have hippocampal damage. So this established that we have a consistent effect and that it's knocked out. Let's take a closer look between these and these, and we'll look at the physiology. Yes?
Just to interpret the EG, since I'm not,
Yeah.
you're considering the top 4 images the same effect and the bottom left 3 the same?
Yeah, and it's not a qualitative judgment, it's a quantitative judgment. So what you can do is you drop it into an ANOVA and you categorize the activity based on the frontal half of the brain or the posterior half of the brain. And that's what this does. Is that topographic analysis found that there's basically that these are all the same and these are all the same, but that these differ from these at a casual
because the first one looks different, but I can see that it's frontal. And
no, they're quantifiably the same. I can say that with great confidence, no pun intended. So this is, we take a closer look, right? So this is our posterior effect. We can say, remember, these should look like this the whole time through, no difference, right? Because consciousness has been held absolutely equivalent. It differs, though. It differs massively in the posterior cortex. And the patients are completely abolished in that, all right? So it actually goes down in the negative direction, and we can see if we plot the 6 control patients, they're all positive, and our patients, each of our 3 patients are down here. So it's not driven by an outlier subject. There's nothing funky happening here. It's a very consistent, robust effect at a group level. So it's not a single case study, anything like that. And this was robustly significant with and qualified by a condition by group interaction. So, and the only difference between this and this is that these patients have hippocampal damage. Okay. So, what if this effect is just too subtle to see? This is a really small group, 3 people. Come on. You guys are all presumably writing dissertations or theses, right? How many of you guys think you can graduate with 3 people in your experimental group? Yeah, I did. But it took me 5 years to test 3 people. So, anyways, what if this is simply due to insufficient power to detect differences, right? That's a very valid concern and criticism. And so what I did was I took my 25 sample group and I randomly broke them down into the similar type of group comparison, 6 to 3. Okay? And when you do that, you see that there is not a big negative blue-looking head here that should be there if it's due to the low sample size, right? All right, what if I got lucky? I picked the right 3 people. Did it 1,000 times. So what you do is you run a permutation test. Run 1,000 times. You create your own null T distribution. And these are our critical T values. This is the T value I found for the real hippocampal patients. So the only thing out there by it was 1 out of 1,000 times. And if you want to bet on 1 out of 1,000 of an interpretation, then I'm happy to play poker with you. Anytime. That would be wonderful. So basically, it's absolutely not due to low sample size. Absolutely validate it and rule that out through an independent creation of our own null T distribution. Larry Squire, the guy who developed the Cohen-Squire declarative memory model system tree that we've been basically bashing for most of the night, National Academy member, president of the Society for Neuroscience. He has a paper that just came out this year on conscious and unconscious memory systems. He says it would be instructive to obtain confidence ratings in association with memory judgments and ask if there is any detectable awareness. Because he said sometimes it's true awareness is continuous and a little bit of it is not the same as a complete lack of it. Right? So what if this contrast that we created, what if it has a little bit of explicit memory just creeping into it that's driving these positive effects in the controls? Right? That's also a valid concern, although we still we can rest assured that the contrast does hold consciousness constant. Let's take a closer look. So basically, to rule him out, everybody in the field likes to rule out Larry because he basically tries to rule out everybody else. He's a very contentious scientist. And so what we did, this is our normal effect I showed you, our positive implicit memory effect when you collapse across all of our confidence bins compared to our patients. Let's take the explicit high confidence recollection trials out. Right? Just use 1, 2, 3, 4, you see the same thing. Now, if the effect is being driven by any type of little tiny, those 5 trials from the explicit responses, and you take those explicit responses out, then that positive effect should go away. So when you take the 5s out, it's still there. When you take the 4s and the 5s out, it's still there. And you can play this game all night, and I did. And we can prove very reliably that is absolutely not due to explicit memory contamination, and it's a truly pure measure of something that is unavailable to conscious awareness. If you were philosophers, you might say, well, we don't know if there's really consciousness that exists, but I can tell you this isn't available to conscious awareness. And so we know that pretty reliably. It is implicit memory, and it is impaired in patients with a hippocampus. So what that tells us is that they are impaired. It's not accountable by factors of sample size, age differences, processing task, anything else. They appear to have intact fluency, that sensory processing, based on this negative going difference. I can explain that later if you want, but it's based on a handful of other studies that have identified similar effects for early sensory processing. And so it suggests a critical role the hippocampus plays in implicit memory. And so it may be that the hippocampus is actually rewiring the mechanisms by which old and new information are processed. And that is happening prior to our conscious awareness. Now, this is actually consistent with some models that Morris Moscovich has put forth from the Rotman Institute up in Toronto. And it fundamentally questions the dogma that implicit and explicit memory depend upon different neural systems. And so we started off with this model based on HM, and we were asked to question everything we were doing by the guy who killed Osama bin Laden. And I was like, okay. So if we can do that, then it seems that if we started with this model and what we've learned now by adding this third dimension of measurements, physiology, that perhaps that's not so true. Maybe there isn't this division between memory systems. And maybe this implicit memory might also be dependent in some ways upon the medial temporal lobe. What those ways are, I have no idea. But it seems that somehow it plays some role. I don't know what the role is. But it's a good starting point to try to figure that out. And so it seems that the brain is remembering what the mind has forgotten. This hippocampus is playing a critical role beyond conscious awareness. And it leads me to suggest that maybe consciousness as a factor is a poor way to taxonomize memory. It's worked up until now, right? But what did our chancellor suggest? Maybe it's time to challenge what has worked. Maybe there's a way it can work better. And with this new information, maybe that's suggests some better ways that it may work. So consciousness might not be the best way to delineate how we categorize things in terms of memory systems. And in a clinical way, some of these subtle deficits may be present when patients present, but not be necessarily evident in our course behavioral observations. And so my conclusion from some of this is basically that the conscious and non-conscious memory systems, they do indeed and may rely upon the same neural structures, i.e. the hippocampus. But critically, they're also still being processed in different ways. So it preserves the view that they are different neural systems. They just seem to be relying upon the same neural structures. So that's a really important point that I'd like to finish on. I'll skip over some of the future directions unless people want to talk about it later. But before I finish, I want to acknowledge both the patients and the controls who participated in this. They gave their time and contributed to science after their injuries. A lot of this was based on collaborators that I worked with at UC Davis. Drew Watros did the pre-stimulus theta thing in collaboration with Charn Ranganeth, Arta Ekstrom, and Andy Younglinks' lab. Preston Thackrell had very helpful feedback on the implicit memory study. He was essential. And collaborators Andy Linus and Andrew Heiser at NYU in New Jersey, supported by a handful of NIH funding and the diversity program from the APA, as well as BBS here. So I'll thank you guys as well for your attention, and I'd like to rest on the notion that I feel that I can have successfully challenged both the textbook of Eric Kendall and answered and picked up the challenge from Bill McRaven. And so I don't think I necessarily need to do more for a day. I feel pretty comfortable resting there, and I appreciate you guys and your attention and feedback. And so if you have any questions, I'd be happy to have them. If not, we can talk about it. Yeah, you can.
One of my earlier questions that is now complicated, I don't know. There are a lot of interesting examples of plasticity.
Yeah.
And so to fingers together and how that changes. Do you have any idea in the patients who don't have a hippocampus,
yeah,
could these regions nearby that we're talking about, could they have a different function than when there is a hippocampus there?
Yeah, so that's a really good question. Basically, the way that I've thought about that in the context of this experiment is, could the effects that I found, that the inability to activate those posterior activations, could they be due not to the hippocampus damage as much, but perhaps some cortical reorganization? Or reorganization of the dipoles of the actual pyramidal cells at the cortex that give these signals. It's a really good question. One of the things, let me see if I can find it over here. This is actually probably one of the control analyses that I did that was necessary for it to be published. So this just got published 3 weeks ago. I was like the only author on. I was sweating my butt off hoping that it would get published somewhere and see the light of day. And I think that your question was one of the things that was necessary to overcome in order to get it to cross that hurdle. And so what we did was to say, now I've shown 2 studies in the same patients where they have big blue patterns in the back of their head when it's knocked out. And it's like, well, maybe they just can't activate that back area. Maybe that there's cortical reorganization or there's something happening in a plasticity way, right? And so what was necessary to do was show that they do retain the capabilities to show these positive red patterns, essentially, in the posterior electrode sites. And so one way you can do this is I tapped into a different contrast, where instead of studying memory and memory accuracy, I collapsed across whether or not they're right or they're wrong in memory. And I just looked at whether they gave confident or unconfident memory responses. And so the idea for that came from prior literature, which has shown that just basically confidence effects are associated with this P3, what they call P300. So it's like it's a positive, the third positive peak. So you might have 1, 2, 3, the third main positive. It's a whole other ERP story. But the idea is that there should be, we can see what it looks like in controls, where you just compare confident to unconfident conditions. And the controls obviously show that confident physiology associated with having high confidence is much more positive than less so. And so when we looked in the patients, they lost the big bump of it. But the most important part, though, was that there was a difference, and it was positive, right? Which addressed those 2 negative blue heads that I think led to your question. And so what we can say here is that it demonstrates the patients are capable of exhibiting these types of physiological patterns. And it's not necessarily attributable to that the cortical things have reorganized. Because if that was the case, then this wouldn't be there either. But it's not 100% foolproof, but it's enough to make, I think, a compelling case. I don't know.
Well, and that's an interesting, it's along the similar line. I think what you're discussing is that the parietal cortex could be doing something different in those patients. And I was even thinking it'd be interesting if you could localize the areas just adjacent to the hippocampus also. And that's part of you saying we don't know what the role is there.
Yeah. Well, the interesting thing too, I didn't want to get too technical during the initial presentation, but the way that ERPs are recorded at the scalp are that they are. It is one of the rules for ERP recording and EEG in general is that it is impossible to deduce the neural generators that contribute to that effect. It's called the inverse problem. And so, and that's okay, but
this is too fast to do with fMRI.
It is.
Can get in there.
Yeah, but we don't necessarily need to because, like I said, those earlier studies with fMRI have found similar things. I mentioned that earlier. Mm-hmm. So, and we already know where their lesion is. It's in the hippocampus. Mm-hmm. So we know that already. But the aspect electrically. The organization of the cells in the hippocampus are sort of organized almost in a circular or a mismatched fashion. So the positive and the negative charges cancel out, and it's called what's referred to as a closed electrical loop. So what we're recording here at the scalp, it should not be interpreted as a hippocampal signal because it cannot volume conduct at the scalp. And this is getting kind of wonky in the physics of it, but
it's down here and you're recording up here.
It's not meant to reflect a direct hippocampal activity. Sure. So whether or not it's a reorganization of the structures right next to it sort of gets mooted at that point because they wouldn't volume conduct either. But what we can say is that the activity at the scalp is a neural correlate of the process of implicit memory, of the processing of implicit memory, and that with damage to the hippocampus, it impairs that processing. So it sounds like you're playing like lawyerly word games and semantics, right? But I would submit to you that words matter. And it's the way we say things in science is really important. And they have different meanings. And so we can link this deficit to the hippocampus by virtue of their lesion, and it's the processing of implicit memory that's impaired. Again, how, why, what form, not a clue, but really cool ideas, and I'd love to follow up with it. And we have some students here that I'm working on some follow-up studies with to try to look at that. Thank you. Any more questions?
Maybe.
Yeah.
Just from my curiosity's sake, what, at least since it's a small sample of patients, causes their hypoxic episodes, and how long since they're middle-aged individuals have they been living with since then? So.
Yeah, great question. I sort of skipped over this, but maybe I should have given it a little bit more attention during the talk. So the